Glad we could catch up after I saw. Well, long story short, like I started off, I've known Zach Lamas for a while, and I've messed with VSH for a while, and I'd
Mhm.
seen some field observations on a colony level about viral infections, mainly deformed wing virus, and he was telling me before that was even published. He's like, "Here's, kind of what we're finding," and I'm like, "No." But like you said, the science was tight.
Right.
Like it was good. So I mean, it gave us a glimpse into what was going on a cellular level, you might say. But then whenever you back out on a colony level, it was the opposite of what I was seeing. And whenever you
Exactly.
said that on Humberto saying, of course I was on mute, but I think I was getting rowdy as a spectator. But it did me a lot of good to hear that because, 10 years ago or so, I was using Tom Glenn VSH
Mhm.
breeders,
Yep.
and I like to experiment. So if I would find a colony that had severe case of deformed wing virus, I would just switch the queen out and not clean them up like you probably should, just
Right.
for observation. And within 6 weeks or a little bit better, I would go back if they took successfully, and it was gone. And so I knew they
Scientists like
had
that.
some type of viral resistance, but I mean, I wasn't even studying entomology at the time, just practical experience. I knew they had some kind of viral resistance, but I had zero data, or I had no idea how they were doing it.
Yeah, it's tough. It's tough to translate the scientific literature and then figure out how does this apply in the field into my colonies. And so yeah, it's important to really read into the methodology always that's used.
It was. And it's great that they did that because
Yeah.
it does give you, I mean, that's happening. That was a, kind of forced trophallaxis, but it's going to happen. And
Right. And it kind of highlights the point about the nurse bees that are performing the hygiene having a higher virus load. And so I think the next question is where's it going?
where do they go?
How are they controlling it? And at least scientifically, that's I think a really interesting question.
Yeah. Did you say you're going to tackle that at some point, probably to see where it's
Oh,
going?
it's on the to-do list.
Yeah. I understand that. Is it like my to-do list for my wife? Like eventually it'll get.
Pages long, right? Yeah.
Yeah. Well, it kind of makes you wonder, obviously they're probably falling on the sword maybe and leaving the colony, as you mentioned, or possibly being shunned, if they know they're sick. That's a possibility. I don't know.
Exactly.
It's
Yeah.
going somewhere because I had actual lab data too to back that up because it was years ago. Randy Oliver was asking for samples from untreated test apiaries and treated apiaries, and I sent him some, which they were probably mostly VSHF1s. And it came back, they tested for A, B, and C strain,
Hmm.
and A and B were BL, below quantifiable levels, and C was non-existent. It wasn't there.
Really.
So I knew they were suppressing it, and those apiaries hadn't been treated in years. So,
Hmm.
I knew it was something the bees were doing, but I couldn't articulate it. And I think you expertly pulled the thread on that. So
Well,
that was
thanks.
pretty
Yeah, I
cool.
have some slides to share if you want to see them. And I threw
Yep.
those in there too, because I think, it's still something that we don't have a lot of data around at the colony level. So yeah, I think it's important still to investigate.
Absolutely.
So
Yeah. You got some slides whenever you're
sure.
ready.
Yeah, so I mean I just put some slides together. I thought I'd share them and I can go through and give a little presentation. If you want to interrupt me in the middle, that's fine. Or, we can just go back through at the end and chat.
Sounds great.
All right. Excellent. Well, first I'll start by saying I appreciate you having me on. It's always a pleasure. And, you know me, I love talking about my bees. So
Same with me. I'm glad you could join me. It just kind of what I had seen from a field observation versus your research. It had a lot of overlap, and you explained some stuff that I couldn't explain. So I'm very appreciative that you were able to join me.
well, likewise, it's great to be here. I always like to just start with a video of hygienic behavior. I know probably most of your YouTube followers are pretty familiar with hygiene, but it's kind of a cool video anyways to show. So of course, hygienic behavior is this ability of the bees to detect and uncap or remove the unhealthy brood from the colony. And so there's been so many different selection methods to select for hygienic behavior. And I think, overall the literature shows that among those methods, there's just a really high variation in the efficacy of these different selection methods for actually being effective against controlling or against varroa, efficacy in controlling the varroa populations. So when I started this work, I really wanted to focus in on like the hygiene communication system. And at the time that I started, a lot of the literature seemed to be associated with the nurse bees and their sense of smell, but not so much on, what was that trigger of hygienic behavior that they were detecting. And so kind of to summarize, several years of work, what I ended up finding with my PhD was that there are these certain compounds that are elevated in unhealthy brood, whether they're varroa parasitized or virus infected. And then those compounds, I call them unhealthy brood odors or UBOs, they can be detected by nurse bees and they trigger hygienic behavior. And I think one of the really interesting things about these compounds and why they were tricky to find is they're actually present on every single bee. So none of the compounds we identified are actually novel to unhealthy brood. They're all present on all bees. It's just that they're slightly elevated in the unhealthy brood.
Wow.
So when they're not kind of in that context of the brood, they're kind of useless and they don't have any meaning. So like I've treated paper towels with these UBOs and I was like, oh, they're gonna, tear them up and remove them. And they didn't touch them, essentially because, out of context, they're pretty meaningless. It's only in the context of brood, which is, why we've started spraying the wax caps, which I'll get to in a minute. So, I'm a scientist, I'm a researcher, and I love fundamental science, asking questions that we have no idea, the applicability. But I also really love when you find something cool, taking that and turning it into applied research. So how can we make this useful? So kind of, we finished my PhD with these incredible findings and we were like, we can't stop here. It's, we're at this really exciting point. So we're like, okay, well, how can we use these unhealthy brood odors to measure varroa resistance? Can we do it? To begin with. And so, again, summarizing like 4 years of work in this little gray text here, we made a mixture of lab synthesized UBOs. So we had some chemists that made these compounds exactly the way that they're found on honeybees in nature. And then we tested a bunch of mixtures and we found kind of perfected this mixture that we call now the unhealthy brood odor assay. So the idea overall of the unhealthy brood odor assay or how it works is you take this spray, it's like a really small volume of this liquid that contains these unhealthy brood odors, and then you apply it to a small area of capped brood, about 45-50 cells in the test area. You return the frame to the colony for 2 hours. You pull it back out, and then all you do is quantify the cell caps that were manipulated. So basically you can count the capped cells initially, and then the cells that remain capped after 2 hours. And so that's kind of the assay, how it works, but really the important thing is what happens next. And that is the beekeeper then has to take that information and then make decisions about breeding, grafting, or management based on those results, and then improve the status of varroa populations in their own apiaries. So just to show how that works in a real life setting. So on the left, I have a hygienic colony, and on the right, a non-hygienic colony. We spray the compounds on at time 0. It actually looks like just a clear liquid, and it evaporates really quickly within maybe 20 seconds. And then the cells look exactly like they did before. 2 hours later, we may see something like this. So on the left, we have, if you look closely, you see there's still 2, I think, capped cells here. So this colony scored a 96%, and this colony on the right scored a 24%.
I've noticed on that slide, if you go back to prior to spraying it, to where it's still capped, some of those caps look like they've been possibly uncapped, recapped.
On the right?
On the left.
Yeah,
Which wasn't
yeah, it's definitely
surprising.
possible.
It wasn't surprising that it was a high test. And I'd noticed in one of Randy Oliver's recent ABJ articles about breeding for resistance, he had one that showed some uncapped, like bald brood, you might say.
Mm-hmm.
Looked at his little circle, there was a few that looked like they were uncapped recaps too, which is awesome because I think they're tied.
Yeah, I actually have some data that shows they are. But it's not always predictable. I've had colonies that I'm like, oh, this is going to score 100, and then they barely touch it. So but I think they definitely are related, for sure, the recapping and UBO. And so I think the important thing here is then, like, what does the beekeeper do with this at this point? So on the left, on the high scoring colony, you may want to limit your treatment. There's anytime you have to put something in your hive, it takes your time, it takes the bees time, it may have negative effects. So if your colony is likely to be resistant, you can limit the treatments that you're putting in. You can choose this colony for grafting eggs, or you could sell it as a breeder queen. On the other hand, if a colony scores low, I would say expect to have to treat that colony for mites, unless you've been selecting for grooming or some other resistance mechanism. You may want to replace the queen or isolate. So, pull some eggs from the colony on the left if you have one that looks like this, and requeen. Or you may want to isolate. I think I'm obviously a really strong believer in the potential for hygiene, but I think no matter, you can have the most hygienic, best colony, if it is absolutely surrounded by mite bombs and these other colonies that you just have mites constantly drifting in. I think even the best hygienic colony can be overwhelmed by mites. So I think there's something to kind of isolating, spreading out your resistant colonies and your susceptible colonies.
Makes sense.
So to kind of test this, we did an experiment, and this is actually an open source paper. I try to publish open source so everyone has access. But this was a fun experiment we ran over 3 years. We had 5 different sites, and we took 72 colonies. None of them had ever been treated for mites. And we ran side by side UBO, assay, and freeze kill brood. I shouldn't say side by side. It was not at the same time, but in each colony, we tested for response to both assays. And again, we're asking the question, does the colony response to these assays predict varroa resistance? So we calculated number of mites per 100 adult bees. And the results, essentially what I want people to take away from this graph, we notice that here we have on the x-axis the UBO score. So we have, in the 2 examples I showed you, 96 would be over here, and I think it was 24 would be somewhere over here. And so what we noticed was around 60% response of UBO. We started to see like these really low mite numbers. And so we started looking at them as like a kind of like the freeze kill brood assay. Marla Spivak has traditionally used 95% as a cutoff. So we started using 60% as a threshold. And I think actually there's some kind of middle ground there as well. But for the sake of this paper, we looked at low and high, and we saw that, the colonies that were scoring high on the UBO assay, 60% or above, had just dramatically lower mite levels than the low scoring colonies. And what was really neat is we saw that the difference was separating colonies that averaged below and above that treatment threshold of 3%, where you pretty much know you have to treat your colonies. We saw, with freeze kill brood, again, we saw a significant reduction, but it just wasn't as drastic. So the colonies that scored high on freeze kill brood, they had fewer mites, but they didn't have, as many fewer. And this is the exact same colonies on the left, that's how they scored on UBO, and on the right, how they scored on freeze kill brood. So we're just, with UBO, better to differentiate a little bit better. And I think this, this explains why we get that variable data on mite control for things like freeze kill brood, probably also pin kill test, things like that.
Yeah. What was somebody had, I couldn't specifically reference this, but it was stuck in my head that, freeze killed brood, or FKB hygiene, doesn't necessarily equal directly mite resistance. Like, like they're better at cleaning out, chalk brood or American foul brood potentially, but like something was missing. Either there's an added.
So I think they're freeze kill brood, pin kill brood, you're really not selecting specifically for hygiene. You're selecting for necrophoresis. You're selecting for removal of dead bees. And I think that's really related to hygienic behavior, but I think it's not quite the same thing.
Do you think that, like, if VSH or whatever we're measuring here, SMR or UBO, is a certain, like John Harbaugh said, it was a multi-trait set. Do you think maybe some of them have a partial gene set, but maybe they're missing? I don't know. I'm just speculating here. Missing something where they're not as sensitive.
It could be. I think, it's so complicated. It's hard to narrow it down to like a yes, no, partial. I think, there's lots of things involved. So with dead brood, one thing we know is that they're releasing oleic acid. So there may be like completely different antennal receptors that are binding oleic acid than are binding these hydrocarbons that are the unhealthy brood odors. So, I think it's just a different thing that you're selecting for. So you may be able to select for individuals that are better at olfaction. So they're more sensitive, they're better at smelling, using dead brood at, selection assays. But you're not selecting necessarily for brood that's better at detecting these specific compounds. Does that make sense? Yeah. So, but I think there's still a lot of questions to answer there.
Oh yeah.
In terms of the genetics. We looked also at overwintering success. So with the unhealthy brood, we saw that colonies that scored high on the assay had 41% greater survival than the low scoring colonies. Again, none of those were treated ever. Same thing with freeze kill brood. It kind of mirrors that those varroa numbers, not surprisingly. So the ones that scored high on freeze kill brood, they were more likely to survive than the ones that scored low, but the difference just wasn't as stark. So we know, hygienic behavior reduces all these different diseases to some degree, but as promised, get into the viruses a little bit. We know there's these really strong links between especially deformed wing virus and varroa. So if you're going to select for these colonies with reduced varroa, of course you should also see reduced varroa associated viruses. But then we see this paper, this 2021 paper that showed the cannibalization of these unhealthy brood is going to also potentially spread viruses to the colony. They're being exposed to these unhealthy brood, especially if they're eating it and then performing trophallaxis with nestmates. So we dove into this question first at the individual level. What's the effect of, hygiene performance on viruses in the individual bee? And so I just ran UBO tests and collected 5 individuals that were actually performing, like I saw them uncapping cells in the UBO assay area. And then I grabbed 5 control nurses from the side. And we looked at 7 different viruses. We never found ABPV, but we found 6 of the viruses we looked for. And, this is preliminary data. I actually have samples in the freezer right now to finish this data set. So I'm really excited to get that data back from a colleague. But we saw hygiene performers have, these substantially higher virus loads than the control nurses. And the effect sizes were pretty big. For example, black queen cell virus, we saw 25 times higher in hygiene performers than in the control nurses. So, but then, as you mentioned at the beginning, how does it translate to the colony level? You have to think of bees at the colony level because they just don't operate as individuals. So, we kind of followed standard protocol to test for colony level viruses. We just ran the tube down the frame and collected 100 random bees, looked at the same viruses and essentially saw the reverse. So we saw the colonies that were hygienic, or, this was based on UBO assay. The colonies that scored high on the UBO assay had significantly lower virus loads than the low UBO. And again, effect sizes were really huge. Deformed wing virus A was 115 times greater in low UBO colonies than in high UBO colonies.
Wow, that's awesome.
Yeah, it was really exciting. And honestly, one of the most exciting things was, like, for example, this Lake Sinai. So, I was expecting this for the viruses that are associated with varroa, but I was really excited that we also saw this effect essentially in some of the viruses that are not associated, not transmitted by varroa. So, Lake Sinai is not known to be vectored at all by varroa, but here it is in, much higher in the low UBO colony. So I think it shows that this assay may be useful for not just selecting for varroa resistance, but also general unhealthy brood resistance, ability to remove unhealthy brood and general disease resistance.
Yes, I think it's going to be big. I mean, I think the timing is right too, because there for a while, like, it seems like everybody just wanted to go the cheap route of which, the chemical treadmill, so to speak, of, well, this is working, why? And now that amatraz resistance is starting to pop up,
Right.
I think the industry is right for something like this to come along and take us to the next level to where we can kind of break away from some of that. Another tool in the toolbox, so to speak.
Yeah, it's another tool in the toolbox. And it's definitely going to take some work from the beekeepers up front, but ultimately the goal here is that the bees do the work for you, right?
Yep.
It's much easier as a beekeeper if you got the bees removing the sick bees instead of you trying to deal with all the problems in the hive.
Yep, absolutely.
Yeah.
And I know Cayman Reynolds is testing some of my stock, which, I've used different VSH throughout the years and have been doing Harbo assays the past couple years, but I was really stoked about this one, in particular too, because one hang up with a Harbo assay, if people use it, is, when was your last miticide application? Because it could give you a false positive or make you think your bees are cleaning it up when you chemically cleaned them up. Whereas
Right.
the UBO doesn't matter if they've cleaned it up or not. It measures their ability to be able to detect that. You know what I mean? So it's kind of irrelevant even. So I think that's a huge advantage with UBO as compared to some of the other stuff. It's way better than freeze killed brood. Harbo and some of the SMR measurements can be cumbersome. Or, like I said, you could get false positives if you're on a treatment regime. So the UBO, even if you're on a treatment regime, you could still measure that and not have your results jacked with as much. So that I thought that was
Sure.
really cool.
Yeah, and hopefully, hopefully one of the other advantages will just be the accessibility of it in terms of time. And with my colleague and I can run through 50 or 60 colonies in a day with UBO.
That's
And
awesome.
there's just no way to do that with with some of the other selection methods where you're looking at mite reproduction and things like that.
Yep.
It's just, they're really effective. They just take a long time. So
Yep, it's true. I with the harbos, like I have a team of people that help me out. Like I entice them with a party or having fun, and they come out and I'm like, surprise, we're doing harbo out. Just kidding. They knew up front that's what we were doing, but they knew we'd have a good time too. But I think
yeah.
that'll definitely reduce the labor input, so to speak, to where you could get results a lot quicker too.
Yeah, that's the idea. I try always when I'm, thinking of technology. I worked before bees. I worked with ceramic water filters in the developing world, and always my focus was like, how can I make this easier for the user? And that's why I started. Initially when I started this, I'd been running freeze kill brood assays in the field, and I was just tired of counting to 160. So I was like, well, what if we just try a smaller section
Yeah.
of brood? And it worked right away. So I was like, all right, not going back up. So it's always my goal to try and make it more user friendly. Because I want it to get used. I think
Yeah.
it's a useful tool, and I'm actually really excited because we're finally at the stage where I've been building this up for 11 years, and now I'm gonna just give it to other people and be like, here, you try it, you tell me
Yeah.
what you think. And so I'm already running a bunch of collaborations and starting to get feedback, and it seems like people are pretty happy with it. I will say I think there is going to be a shock, because I think people that are used to testing with freeze kill brood or pin kill test are used to getting higher scores, and so they run the UBO and they're like, well, none of them tested high.
Yeah.
It's just a more sensitive test. So I think people are going to have to build up their UBO scores, because it's a more sensitive, more subtle test, but I think it's doable.
Yeah, and I think that's really important that you just mentioned that too, to encourage people, because I know like one of the collaborators of yours, I don't know that he may have been hygiene testing, but I don't think he was specifically measuring VSH or anything. And I was worried that he might feel defeated potentially by, I mean, I know he had some that scored high, but do you know what I mean? Like if
Mhm.
you walk into a random population of bees and start UBO assaying them, you're probably going to be disappointed with the vast
Yeah,
majority of them. But it's a start,
right.
and you have to build from there. And like even if you can, part of it is what level or what percentage of your brood nest expresses that. So that's a factor as well. It may be a low concentration, but like you said, if you start picking those as breeders and grafting out of them, you'll
Right.
start to
You
see
got to
that
start
increase.
somewhere. Exactly.
Exactly.
Yeah, I think in my experience, if you have kind of a population where you haven't really selected for hygiene at all and you test them, maybe one in 25,
Mhm.
um, 1 in 30 colonies is gonna
Mhm.
score high. It's there naturally in some colonies.
Right.
It's just if you haven't been selecting for it, it's hard to find. And
Yeah.
it's correlated with freeze kill brood. It's correlated with pen kill, the UBO responses, but it's not the same. So I get
Yeah.
high freeze kill brood that score low on UBO, and high UBO that score low on freeze kill.
Yeah.
These
Wow.
are a mess.
They are, but that's why I'm an ADD kid, and if I mastered it, and I would get bored with it and quit. So there's so many variables. I think that's what keeps me interested. Is you can keep pulling the thread, but it seems like you're never going to get to the end of it. But I
Right.
was curious too, because you compared freeze killed brood overlap with UBO. I'm curious because I think there'll be a more pronounced, I could be wrong, more pronounced overlap depending on how they were testing it with like VSH SMR. I think if you
Mhm.
have a VSH or SMR tested stock, I would predict, could be wrong, that you're going to have much more consistent or higher UBO scores
Yeah.
because there's got to be some overlap there. Because I was using, and I'm assuming they were using Harbo assays way back in the day, Tom Glenn's VSH stock, because they were viral resistant as can be. That's how I
Mhm.
figured that out originally. So where that there's got to be overlap there, I feel like.
Yep. Yeah, and we have some data starting to come in to show that.
Awesome.
Yeah.
Yeah, I'm
Oh, so
anxious
much
to
to do.
try it.
Got to write those up.
What's that?
I said so much to do. Got to write those up.
Understand.
Um, yeah, so I mean, we kind of already talked about the, this paper. But I just want to make the point, this paper, like I said, really solid science, was consistent with some of our findings, like the finding of high virus loads in the hygiene performers, but it wasn't consistent with the picture when you back up and look at the whole colony.
Hmm.
And so I just want to kind of reiterate that when you're, as a beekeeper, trying to interpret the science, just make sure you dive into the methods and understand how the study was done.
That's true.
In this case, at the colony level, there's probably something going on that wasn't going on in those cages. Like you mentioned, social exclusion, maybe they're even full from cannibalizing sick brood, that they're not asking for trophallaxis and they're not giving it to nestmates because they, somehow perceive they're sick, they're self-isolating. We don't know, and I think this is a really interesting topic area for researchers to take on.
Hmm.
But we know that something's going on.
Definitely.
Yeah, it just somehow these performers are like, I think of them as like virus sinks. They're just taking in all the viruses of the colony and protecting the colony.
Hmm.
I think one interesting question, it's probably likely that they're getting the viruses from the, cannibalization. I mean, I'm sure that that's at least one way they're getting them, but it's also possible that, bees that are already somehow sick are the ones that designate themselves as hygiene performers because they already, have viruses. So that's another thing I think would be pretty neat to untangle. Is it just because of their task performance that they have these virus loads, or is it actually somehow they become that task performer because they already have a high virus load? Be cool to see.
Absolutely.
So, one thing I've been focusing on the last year or so is like, um, hygiene is pretty cool. I think it
Oh,
seems really promising. So, it kind of confuses it, at least it did, why don't all beekeepers, why isn't everybody all over this? Why doesn't everybody just keep hygienic bees? And so I've been talking to a lot of beekeepers, and I think kind of the top reasons that I've found from beekeepers of why they're not keeping hygienic bees, at least regularly, is for one, some just don't know about hygienic bees. I think the word hasn't spread as much. I think it's starting to. It's starting to pick up. Like you said, we're kind of people are moving away maybe from the compounds, the chemicals, some, and more interested in natural processes. Also, there's not always access to hygienic queens, so even if you know about it, you may not, know a breeder. Um, One of the big issues I think is there's a high cost of the high performing queens. So, I've seen like hygienic queens for sale on Craigslist, but are they really hygienic?
Right.
If they are, you know, they probably should cost several $100s
Yeah.
because
Yeah.
somebody spent hours, going through and looking really closely at mite reproduction or colony mite levels, hygiene, some kind of really labor intensive selection. Another thing I've heard a lot about is like, well, what if I'm willing to, throw $300 into a queen? what if she absconds or something happens to that queen? Do I just lose the trait? And so this past summer, I did an experiment looking at heritability of hygienic behavior.
Yeah.
And so I just wanted to show you some of those results here.
Yeah.
Do what?
That's awesome. I was hoping you had that. That's pretty cool. And I had some thoughts on that too, that might be from a different angle.
Awesome. I can't wait to hear them. Yeah, so essentially what I set up was a high hygienic apiary. So I had average UBO score in this apiary was 85. And then I had a low hygiene apiary. The average UBO score was 29. I didn't have any low scores in the high apiary, no high scores in the low apiary. And then they were separated from each other by about 5 miles. Neither one, of course, was isolated from other beekeepers. Can't do that. Don't live in an island, but
yeah. Sounds good though.
Life goals, right? So from each apiary, basically I made 2 splits from each of several different colonies. And then I just translocated one of the sisters and had her mate in the other apiary. So essentially we have at the end 4 different types of nukes. So I had a nuke that was mated. So she was grafted or came from an egg with a mother that had low hygiene. And then she was mated. This was all open mating. So she was open mated in a low UBO apiary. I had the ones that had low mother's hygiene, but they were mated in the high UBO apiary. High mother's hygiene with the low mating apiary, and then high mother's hygiene with the high mating apiary.
That's a really cool experiment.
Yeah, it was really fun to do. I did over 2 years just because it's pretty labor intensive to do. And was like one of 13 objectives for last summer. But it was really, really fun to do and really cool to see, how clear the results were. So we ran, essentially what I did was I ran UBO tests then on each of these daughter colonies at 6 weeks post queen emergence, 7 weeks, and then greater than 10 weeks. So it was like 10 to 14 weeks, depending on when I actually got the queen to take. But essentially I wanted to test them all, not necessarily at the same age like I did the first 2 tests, but at the same day, just to make sure there weren't, day effects. And so this is the results, and I've taken the kind of the numbers off because this isn't published yet. Again, on the to-do list. But what we found was, the mother's hygiene seems to have the biggest impact. So here we have the 2 colonies. Can you see my cursor here?
Yes.
Okay, so on the right we have the 2 sets of daughters that came from the high UBO mothers. And so you can see that they're higher than the 2 sets of nukes that came from the low UBO mothers. But in both cases, the mating yard had an effect. So if they were mated in the high UBO apiary, they had a higher average UBO score than if they were mated in the low UBO apiary.
That's awesome.
Yeah, it's fun to see. And, I think it ties in really well with this last point about why beekeepers don't have hygienic bees is they've either had bees that were sold as hygienic, or they've heard about people who have that, just didn't perform as hygienic queens. And so, this lowest score in our high group was below 40%. So, I just think it really shows like you can try to control everything. And, unless you're doing artificial insemination, which, is great, but also expensive and a time sink, you never know what you're going to get. And so there's just a lot of variability. I'll also say, I was actually just looking at this morning, this high group, the daughters actually scored on average 16% lower than their mothers
Interesting.
And on this one that was the high mothers, but made it in the low apiary, it was like 30% lower than their mothers. So, you know, if you're gonna select for UBOs with open mating, you really gotta have a lot more daughters than you have mothers, because you want to be able to scan through those daughters and pick the best ones. You know, it's a genetic mashup out there in those drone congregation areas. So, you know, you don't know what you're going to get and you have to test for it.
Mm-hmm.
So what we learned from this, you know, I didn't show all the data from, that was actually the 7-week data. 6-week data, 7-week data, and greater than 10 all looks exactly the same in terms of the relationship, you know, the pattern, but the you know, the variability is just huge at 6 weeks. So definitely wait at least 7 weeks post emergence, 6 weeks laying before you start testing for UBO. You can't, you know, you can't throw a queen in there and expect to be able to test her right away, because you really need those older workers to clear out before you can get reliable scores. And, you know, 10 weeks, of course, even better, small error bars, but you know, I think it's totally fine to test it about 7 weeks after emergence. We saw again that the mother's UBO score is important, but the drones that the daughter mates with also have an effect. And so make sure if you're selecting for hygiene, your neighbors are too, essentially. And I think just, you know, in general, regardless of where you pull your eggs, where you mate, you got to test your daughters. If you really want to know that you got hygienic daughters, you have to verify. So just briefly, like the importance of the work, I think, you know, like I just said, you can use this assay to verify hygienic daughters. I think one really exciting thing about the assay is that it's going to facilitate local selection. So I'm working with a group up in Vermont. You know, they want to select for varroa resistance, but when they bring up these breeders from South Carolina, Georgia, Louisiana, that have been selected for VSH, they lose some of their winter resistance. And so I think it's really, you know, bees do locally adapt. Even here in North Carolina, we have, you know, 3 pretty clear climate zones within one state. And so it's really nice to be able to say, hey, look, these are my bees. They're, you know, they're good for the reasons that I want them to be. You know, they're gentle, or they're high honey producers, or they're winter tolerant, whatever it is that you've selected for in your bees. And then you can, within that population, start selecting for hygienic behavior, varroa resistance.
Absolutely. And I think in that would also, you know, I know you've heard Sue Kobe or some of the ones in the past say that, you know, we have an issue with sex alleles, or basically inbreeding, or a lack of. Well, if everybody's not getting their breeders from 1 of 2 sources or 3 in the US, and it's more of like I use a micro brew, like beer model, you know, instead of everything coming from, you've got your own little craft, you know, your own locally adapted stuff.
Grow your own hops, right?
What's that?
Grow your own hops.
Exactly. Well, if you're doing it yourself, well, then if somebody's breeding program has inbreeding, well, then you've got way more of a pool of locally adapted stocks to make your crosses, get some sex alleles back in, and start selecting again. So,
right. And it's not to say don't bring in outside bees, because I think you have to bring in the genetics from outside occasionally. You know, but if there's other people, like you say, if there's a lot of other people selecting in similar climates to you, even if it's across the country, if they got similar weather, similar traits that you're looking for, you can exchange with them, and you can hold on to those important traits.
Definitely. And like coming from California, that's where Tom Glenn's bees came from originally. And the best ones I had were F1s, where they outcrossed with what I had.
Right.
But they had super high VSH expression, because that was the problem with some of the local stuff I had, is they didn't have the level of mite resistance that they needed. So then you've kind of got a local blend of that, and not
that hybrid vigor.
hybrid vigor, you got heterosis going on, so they're strong. They've got the mite resistance, and they just do so much better that way. And then you can, this is the thing, and that's one of the main things. I'm glad you talked about the heritability of it, because every time I talk about VSH or hygiene, that's one of the questions that always gets asked is how long does it stay? You know, my answer is always it depends. And you covered it. You know, are you actually selecting for it? Because if you actually keep selection pressure on it, it could not go away, period.
Right.
But if you're
It can
just
increase.
guessing,
Yeah.
yeah. So that's a huge part of it. And the drones, because, you know, they don't have a father, only a grandfather. So they're a recombinated set of their mother's DNA. And like you said, if you're testing those and grafting out of those, you can totally keep it and have your own local hygienics.
Right. And yeah, and so I'm hoping some communities, you know, some groups of beekeepers from the same areas will kind of take this tool and use it as a community and see what can happen. You know, I bet it happens a lot faster if people can, you know, can work with their neighbors on it and select for
this. And as far as that goes, I think one word of advice would be keep it simple. Like everybody has certain things they want to breed for. Like if you just say this is the only thing we're really looking at, it's UBO. Like whatever else, like they need to be productive, they need to be relatively gentle, but this is the only thing. You know, if you get multiple traits, you know, then you'll start having disagreements and things of that nature, because, you know, we can't even agree on whether to use screened or solid bottom boards. So that would be my only advice, is absolutely do a little local cooperative, but keep it so simple that you're united. You're united for the cause.
Yeah, great advice. You got to focus on the end goal. I mean, not let the other traits slide at the same time.
Right.
But yeah, I agree. I think, you know, comparing this again with freeze kill brood, one other advantage that I just want to point out is I think it really improves our resolution of hygiene. So there's not a lot you can tell in the 95 to 100% frame. You know, it's really just like a binary high or low. But I think with the UBO assay, we have a little bit more flexibility. So, you know, this is just an example. But maybe those colonies, you know, if your mother is 60%, maybe the F1s are going to be really not so resistant. You know, you really lose that trait pretty quickly with grafting and the F1 generation. Maybe if you're using natural mating, then you really want to go for the higher end. So you really want to focus on those mothers that are 95, 100%. But if you're doing insemination, maybe you want to go for the middle ground, you know, because you know you're going to get those drone, those semen that you want from the drones that you want. So, you know, this is just an example. You know, don't take these numbers to mean something actually, but I just think that it gives us a little bit more resolution in terms of, you know, looking at, okay, these are relatively resistant, these 60 to 100% scoring colonies. But within that, what do we see? And how can we use those high scoring colonies differently to achieve different goals? In terms of just beekeeping management, as I mentioned before, I'm going really for accessibility. So what we're trying to put together now is just something that is really beekeeper friendly, you know, whether you have 2 hives in your backyard or you got multiple apiaries with hundreds of thousands of colonies. We want something that's easy to use, quick to use. I think it can be useful for, you know, of course, for the breeders, but also for people that want to compare queen breeders who want, you know, who have 5 colonies in their backyard and they lose a queen. Where do they pull their eggs from? You know, so I think even on that small scale, you can start increasing your resistance, even in a small apiary. And then, like I mentioned before, and I'd love to hear your take on this, but I just see some potential in and trying to separate the susceptible and resistant colonies and avoiding some of that mite drift that can cause problems.
Completely, from a breeding and a management perspective.
Yeah,
And
exactly.
and there's a lot of people that, there's kind of some confusion on, a lot of people, if they're raising queens, they consider themselves a breeder. That was myself, which I wasn't breeding. I was just a good queen rearer. But there's a lot of people that don't necessarily know what to do next or what has the most meaning, because you can get lost in selection and data. Like, you
Yeah,
can't select for everything, or you're
right.
selecting for nothing. You know what I mean? Nobody stands out then. But
So
I think
what do you select for first? Do you take like your best honey producers and your most gentle colonies and then select for varroa resistance, or the other
pretty
way around?
Pretty much. I make a whole bunch of F1 colonies, and then I don't put anything on them. I just let Mother Nature start sorting wheat from chaff. So you've got the natural selection element. And then whenever they come out of winter, they have to do at least one full cycle. Whenever they
Hmm.
come out and the nectar and pollen turns on, like, I want them to be super responsive. So that, to me, that's productivity. If you
Sure.
have nectar and pollen coming in and you're just kind of stale, that's a cull. But if they take off, like they're gonna put up, you've got brood to split with, you've got big colonies to raise queens from, or if you want to make honey, that those are productive. So I go through those, and then, like, the ones that stand out the most, those are the ones I VSH test. Because I know Zach Lamas had told somebody, like, he VS or he SMR tests all his colonies. I'm like, no, not exactly. That's pretty labor intensive. I only select potential breeding candidates.
Right.
And I don't.
The cream of the crop.
Only the cream of the crop. Yep, exactly. And I try to keep it relatively simple. Like, if they're completely overly aggressive, they're a cull. But even if I have sometimes I'll have an F1 cross. Like, I had, John Harbaugh F1s, and I would inseminate them with my high scoring colonies. Some of those would be a little bit aggressive. So
Hmm.
I've gotten to the point to where I'm not going to bring anything else in, because I was in John's breeding group, the stock trade and stuff. I'm not going to participate. I'm not going to bring anything in, and just because I basically run like a variant of a Page Laidlaw closed population breeding model with
Hmm.
instrumental insemination. And so I just want that to run its course and see what it produces, because, like you were saying earlier, if you bring in stock, you have something that does really well here and you outcross it, you know, you'll gain one thing, but you'll lose something else. Like, I gained my VSH scores were getting higher, but then I started getting some annoying aggression in some of those hives. So I don't want to bring anything else in and just let the model keep running, but I want to add UBO as well. A, to compare it with just a Harbo selection, and B, because I think there's so much overlap there. I'd like to see how it pans out. It may be where I just ditch the Harbo and just do nothing but UBO, because I feel like you said, it's showing obvious mite resistance, and that's the main thing I'm worried about.
Hmm.
Mite resistance first, productivity second, because if it's mite resistant but it never leaves a single brood box, it's useless on the commercial
Right.
scale. I'm like you, I want something that's practically applicable. So that's what I do. And I learn something new every year, and I change something every year, but I've been at it a little while.
Well, that's a good sign.
Mhm.
With beekeeping, you can never know it all. I feel like the further in I get, the more confused I am.
Yeah, the more you know, you don't know.
Exactly.
Yeah.
Oh, 100%. Yeah. With bees, at least. I mean, they're fascinating.
Oh yeah,
I love my
absolutely.
job.
Yeah, I would like to do it full time, but I don't know. That's another thing, like we were talking about, why doesn't everybody use hygienic breeders? Or, I think there's multiple reasons, and you touched on a couple of them. I think also, whenever they first came out, somebody's like, well, what if they're too hygienic? That was kind of I look at it as an old problem. Like, whenever the VSH program first came out, they had some that were kind of psycho hygienic, but they
Yep.
culled those. And then I haven't seen that issue. I'll have some that
Yep. Exactly.
you'll get some bald brood going if they have a stressor, but overall, I've got large colonies that are full of brood that'll test a 4, on a harbo, but they're not crazy hygienic. Matter of fact, the brood patterns look better than, a lot of stuff I see on Facebook.
Hmm.
So
Yeah, I totally agree. I saw some of that in like, I don't know, 2014, 2015, and I haven't seen it since.
was it like
And it was
hyper involved brood or hyper?
no, pretty much over like obsessive compulsive
Oh,
hygiene and, just removing everything. And it was those early VSH, but I really haven't seen it since then. And, it
yeah,
comes back to that scale though. Like, if your 100%ers are all obsessively removing colonies, then removing healthy brood, then just, go to your 80%ers and,
yep,
it
yeah.
just gives you that flexibility. But I haven't seen it in several years, so
I
I'm
wonder
more
too.
worried about mites.
Oh, absolutely. And I also wonder too if, like, obviously there's varying levels of sensitivity. Like if they get exposed to like chemical exposure and everybody's sick or something, you know what I
Mhm,
mean? That may be one you want to keep potentially, but I would absolutely use caution. But keep them for observation because I've had
mhm.
some that like had a high level of uncapping, at least maybe on one frame, and I would go back later and it would look normal. So, you know,
Mhm.
who
Yeah,
knows?
and I'd say, you don't necessarily want to take out if a colony doesn't score high on UPO, but you like the colony, keep
Mhm.
the colony. Don't,
Yep.
don't trash a queen because they don't score well.
Yep.
You
Exactly.
never know. So I've had like ankle biters that, were really good against mites. They had really low mite loads, but they scored 0 on UPO. And there's other mechanisms of resistance. So this is just one, like you said, one tool in
Yep.
the toolbox.
Yeah. And I've
But
kind of touched on that too, with coming from an entomology background, we get IPM drilled in our head, and it's like everybody just glosses over host resistance. Like we go right for other stuff, it seems like, but I feel like that's coming back around now. So I
mhm.
think that this is good timing for that. But,
Yeah,
if
I want some groomers crossed with hygienic bees.
only one, it may not work out this way for you, but I mentioned earlier, like if I make an unrelated cross, like I take something I instrumentally inseminate, like I had my stock and I instrumentally inseminated with some Purdue biters,
Mhm.
and like there was a portion of them that were just wicked, like, and I sold a few of them, and I told them,
Just
yeah,
the defensiveness. Uh-huh.
yeah, they were hyper aggressive. Like, I mean,
Right.
they hated hive beetles, they hated mites, and they hated beekeepers. But the thing is I didn't have enough time. Somebody would, I could figure out how to get paid just to breed bees all day. I could have sifted through that because some of them were great, and I know some of them they had it all. But it was only a portion of them that were gentle. They probably had high VSH levels, and they also had high, groomers in the brood nest too. But
Right.
I haven't gone back. Maybe I will. We'll see.
Yeah, I mean, if you could attack those mites from the adult bees and in the brood at the same time,
You got it made.
oh, you got it made. But
Yeah.
yeah,
But if
like
you
you
have to
said,
pick what you're selecting for here, I think like on the, if you list out whether it be VSH, SMR, whatever that equates to, hygiene, allo grooming, auto grooming, apoptosis, like you list out all the stuff that is a resistance mechanism. I think the targeting reproductive founderous mites in the brood is like the root of it. Like I
yeah,
think it's superior. If you have to pick one, I think it's that one because not only does it address the root cause, which is mites reproducing and making more mites? They directly target that, but they also have extreme viral resistance and, just general unhealthy brood, chalk brood, American
yeah.
foul brood. You don't see that. So like it's so multifaceted that's why I'm really glad this assay has come out, and will help push people in that direction because it's so multifaceted as
Agreed.
far as
Yeah, and
great.
when I compare mite loads on adults and in the brood, I typically see about 3 times as many in the brood.
Mm-hmm.
That's where they want to be. If there's brood around, that's where they are. So you're also just going to target a higher number of the varroa. But yeah, it's more, one of the cool things I think is at least one of these compounds that we're using was found to be elevated on unhealthy ant pupa that was selected for removal. So this is not like a varroa specific thing, you know. I think it's been, these hydrocarbons have been essentially co-opted by social insects to
Yeah.
just trigger removal, or to, you know, to signal general unhealthiness. And so that's one thing I love about it, is that I think it really kind of covers a lot of your bases in terms of diseases. There's a lot more research to be done to show it for sure, but yeah.
It's pretty exciting stuff. And it makes sense if it's in, you know, honeybees, it's in other hymenopterans too, you know, I mean, that makes sense.
Right. Yeah, and I just want to make one more point on, you know, potential significance.
Mhm.
My love and my focus is on honeybees, but I've started to pay more attention to native pollinators too.
Same
And I
here.
think when we're selecting for varroa resistance and selecting for disease resistance, we really have potential to decrease the spread of like these diseases to native populations of pollinators.
Yep.
So there was a study that showed from 2022 that showed like bee pathogens were found in 42% of bee species and 70% of flower species.
Oh, wow.
So like, you know, native pollinators are sharing these resources with honeybees. And I feel as a beekeeper, I have a responsibility. You know, we don't know what's going on with the native bees because we're not managing them. So I feel like I have a responsibility to keep my bees healthy, my honeybees healthy, because that and essentially is going to help protect native pollinators.
Absolutely.
So
That's pretty cool. Well, I think it was partially entomology degree that I was working on that, you know, kind of shifted my focus to native bees, but it was also like I participated in like Missouri Bumblebee Atlas. That was mind blowing because I thought I knew bumblebees too, but I didn't know anything. Like I learned a lot of how many different species there are. And then you get into like Melissades, like longhorn bees, and
yeah, they're I'm just starting to get into native bees, and they're so cool. I love that down here in North Carolina we got those metallic green sweat bees.
yeah.
They're
Is that like
beautiful. They're just so,
Algochloris or something? I think is maybe
yeah, I can never
pellet
remember
sweat
the,
bees. But we've got a few of them around here too, like little bitty tiny ones, and then we've got some larger ones too.
yeah. So yeah, I feel that beekeepers should, you know, I mean, you can put chemicals on to kill the mites at the end of the season when the populations are up, but the viruses are already there if you're waiting, and they're already out of impacting all these solo bees that are like hibernating over winter, one queen at a time. And you know, I just think that has a huge impact on making native pollinators really vulnerable if we're
I've
not.
never even thought about that aspect of it, to be quite honest, because there is some overlap. And I know there's occasionally I'll hear someone say, you know, like everybody wants to look at things black and white, which I think is really dangerous. But they'll look at native, non-native, all bad, native, all good. You know, I'm like, it's not that simple.
Yeah.
But you know, there's some debate over forage and whatnot, like how much honeybees negatively affect native pollinators. Like if there's nothing to eat, there's going to be some competition. So,
Sure.
and there was one podcast I'd just done, like if you want to help the bees, you're not a beekeeper, you're not breeding for better bees, like plant flowers. You know,
Yeah.
we did 14 acres here at the farm. That's just nothing but diverse wildflower habitat. And I
That's
love it. Just poking around out there, it's like a jungle with all kinds of different species, and it's pretty cool.
amazing.
So yeah, breed better bees and plant some flowers.
Absolutely. 100%. I'm with you. I just kind of wanted to summarize what we know so far. So it was actually kind of nice for me making this slide, because I was just like, oh man, what do I know for sure? What do I think? And what do I have no idea about? So far with UBO, I think we have pretty concrete data showing that, the high UBO is associated with the lower varroa infestation, the higher overwinter survival. Now we know the higher F1 queens. And actually that one should also have the not published yet star on it. And the long-term memory of UBO. So this is a study that I did actually like, I don't know, 2-3 years ago. And I really need to get published, but I found that the colonies that scored high on the UBO assay were better at remembering essentially these UBO compounds 24 hours later. So I did this really fun, the proboscis extension reflex, where the bees, if they get a high enough sugar stimulus, they'll stick out their tongue or their proboscis. So I did like a classical conditioning experiment, Pavlov style.
Kaira's bees.
Yeah, so I trained them basically to I would put the UBOs on their antenna essentially with a little glass capillary, and then I would reward them with sugar to the point where I could I could just touch them with the UBOs and they'd stick out their tongue, right? They would associate, they'd be like, the sugar's coming.
That's awesome.
And it was really cool because I found that the ones that scored high had a better memory of those UBOs 24 hours later.
Interesting. That's too cool.
Yeah, and I think it's actually kind of associated with something down here. This last point in orange is I've been working with just small preliminary study on odor and binding proteins with the Foster Lab up in Canada, and we've found some candidates that we think are there's greater expression of certain odor and binding proteins that are associated with high UBO colonies. And I think it's going to come back to that difference we talked about before between like dead brood smell and unhealthy brood smell, and the different compounds that are related. So that's something that we're just kind of starting, and we have some really early data on, but we already see some significance. So I'm excited to explore that. We have data, I'll just go up from the bottom. We have significant data correlating recapping behavior and UBO response. Still, so this is like, I say preliminary because it's been one study or 1 year only of a study. We're still analyzing data. But the data's starting to come in. We're starting to see correlations between VSH, like that SMR score,
totally,
and UBO. And then I just can't wait to get this virus data in to see if results from a bigger data set are consistent with that exciting data that we saw before. But I expect they will be, especially for the varroa resistant, the varroa associated viruses. And then there's some things we don't know yet. We haven't looked at chalk brood, we haven't looked at EFB or AFB, other diseases. So there's still a lot to do. Keeping me busy.
I'm curious about that too, like where you put VSH based on my non-reproduction. There's like, I've been obsessive over it for a long time, but like the overlap I see with that was the viral reduction key. So that one was big. And then like, VSH is a type of hygiene, which is why it was renamed from SMR to VSH, but they target varroa specifically, which lowers mite levels. Well, the thing is like that's where I was looking at freeze-killed brood, UBO, and VSH. I'm like, these two are very similar because this one may or may not predict low mite levels or extreme mite resistance specifically, but these two, there's something going on there. You know what I mean? That's why I was looking forward to adding it too. And don't let me forget, I need to go back and add that link, because if someone listens to this podcast and you're interested in getting UBO, it's really close to hitting the market. And there is a list that you can sign up for, which I already did. And I know several friends that have too, to where you can get on a wait list. So I'll add that to the notes as well. So if you're really interested in this, it's coming.
Yeah, I appreciate it. We're hoping for spring of 2024. That's the goal.
Awesome.
So we do, and I actually on my last slide I have our website and QR code that you can scan if you want, just
Beautiful.
to link to it. But we are taking pre-orders. We're trying to get a sense of how much of this stuff to make and, you know, where people want it and, you know, what time of year they want it. So we know how much to make and put together. So the deal with the pre-order right now is, you know, it's completely non-binding, but our plan is to kind of reach out to the people that made pre-orders first and fill those orders as like a priority
Sweet.
right before we launch. So that's
Awesome. And you'll be at ABF talking about it?
yeah, I don't have a talk, but I am planning to go to ABF.
Will you have a booth or anything, or just if you're interested in it, find you and
Find me, let's talk.
okay.
Yeah, I'm going down to meet with some breeders about you know, a potential project we're gonna do. So some plans for the future. But I figured I'd go down and meet with them, and anybody else that wants to talk, find me, and you know, I'll be there. You can email me beforehand so I know to look out for you.
Awesome. And your business partner will be at Hive Life, correct?
Right, yeah, they're scheduled at the same time, so we just had to divide and conquer. So Phoebe Snyder will be at Hive Life and I'll be down at ABF.
So if you're interested, find one of you two.
That's right.
And I think that they won't be on the same date for 24. What was that slide just before that with a lot of uncat? Yeah.
Well, yeah, I was gonna skip it.
What,
All right, you call me back. No, I just wanted to put a reminder, like, you know, it always makes me nervous that people will get so excited about UBO and only focus on it.
Yeah.
I don't want people to forget about the other important traits.
Yep. Agreed.
Yeah.
Which I'm glad you asked about my program, because I guess to articulate it, the mite resistance is actually the last hurdle, you know, because they have to go through you know, natural selection. They have to be productive. They have to respond to nectar flows. They have to not tear my veil off. And then the last hurdle is actually mite resistance or SMR measurement. But
yeah, I think that's a good way to do it if you can, especially if you're able to increase the scores over time.
Mm-hmm. And that'll keep you from having to test everything, because you know there is some level of time and expense to it. So you're really only going to want to test what you are really wanting to breed from.
Absolutely.
Most likely.
Yeah, so you know, moving forward, I am like neck deep in collaborations, which is an awesome thing. Like I said, I'm just really happy to be getting this out to other people to try it and give me their feedback. I want to keep exploring virus dynamics in the hygienic colonies. We're looking at these odorant binding proteins. And I mean, right now, my biggest focus is just getting this thing out there. So I don't know, spring 2024 seems like a long time from now, but there's so much to do to get there. We're still working on this applicator. We're making progress. But we also want to just have some kind of background information on environmental effects. So put together like, you know, a best practices manual essentially for people. So they know, you know, can I use my smoker when I'm running the assay? If I got a screen bottom board or screen top, does it impact the assay, you know, because there's more airflow. I saw this year, like, I think nectar flow just has a huge impact on the assay results. You know, so we see you know, from April to June, we see these great scores. And then when the dearth comes, the scores just drop. But this past year I saw when I was feeding, you know, in August when there was no natural flow, but I was feeding, the scores jump back up. So, you know, that's all just anecdotal. And so this coming summer, I really want to look at nectar flow and in a controlled experiment and see, you know, how it affects the scores.
That is so crazy. Because I mean, if you notice, you know, like if you look at a calendar, you know, and associated with mite levels, typically, you know, fall, September, October, or even August occasionally, it could be tied to nutrition. Your mite levels go through the roof. I was just talking about that with somebody else, and that's really interesting. So do you?
Yeah, and Marla Spivak had actually told me that about free scale brood. years ago. I mean, she was on my PhD committee. She's been pretty fundamental to this work too. And she told me years ago that she had noticed that basically when the flow is on. And, you know, at the time I kind of attributed it to they're clearing stuff out quicker because they want somewhere to put the honey, but you know, they're not always nectar bound. And I think it may have to do with, you know, priorities and, you know, if they got enough energy to perform hygiene, they'll do it.
Interesting.
Yeah, I don't know yet, but I'm really interested in diving deeper into that in a more controlled way than just, you know, noticing it in my apiary. So
yeah, I'll look forward to seeing you pull that thread. That's pretty
yeah,
awesome. I mean,
more to come on that for sure.
great.
Tons of people have been involved in this work, so I can't end without, like, you know, mentioning UNC Greensboro, my alma mater, USDA, NSF. We've done the I-Corps program there. I mean, NSF has been absolutely critical in getting this, kind of helping us push this from the lab to the market. I never thought I'd be a business person or an entrepreneur, but you know, they've done all this training and they're going to help us get there.
Awesome.
Project APISM, of course, has been like fundamental, as has the USDA and like a lot of the PhD level support. And even since then, North Carolina Biotech, we got a North Carolina matching grant from the small business program. Got a shout out to Inside the Hive. And then more recently, I've started getting support from like local beekeeping groups. You know, so we're really trying to push this forward, and Ape and Bees, H&S Bee Supply, and Surrey County beekeepers have all given like donations to kind of help us move this forward more quickly. So, you know, every little bit helps, and we really appreciate the support we've gotten from everyone. And then here's your QR code.
Okay.
Fancy new tech, right? You can also just go to opterabees.com, but you can scan. This will just take you there. We're still, the site's still under construction, so you may see a couple pages that aren't done yet, but we're working on it, and we are starting to accept those pre-orders, like you mentioned. So
awesome, and I'll put
check
a
it
link
out.
to it too in the notes.
Okay, great. Yeah, come check us out, and you know, if you have any questions, there's our emails on there. Feel free to reach out and tell us what you're interested in. You know, we'll launch in spring 2024. We'll launch in the States, but we want, you know, if you're outside the States, we want to know where you are, because you know, we'll probably have to target one region at a time when we expand beyond the States. So we want to know where people are that want it. So let us know.
Awesome. That was great.
Super. I'm gonna stop sharing so I can see your face again. There we go.
Well, I can't thank you enough for that. Like I said, just seeing the whole thing play out, you know, like talking to Zach, you know, and him telling me kind of what was going on before they published that study, which, like you said, was good science, and it was a tight study. But whenever he told me that, I was like, no. I didn't say that because I didn't want to discourage him, you know, because I knew the science would be good. But I was like just kind of distraught over it because it didn't match what I'd seen. So I'm like, well, maybe there's a disconnect between, I don't know. There were so many thoughts going through my head. And then Humberto, Inside the Hive TV, if you haven't seen that interview with Kaira, you should go watch that. And then he's expertly put that out, you know, to get everybody worked up, and then waited a bit and then did the follow-up with you. And whenever I saw that, I was like, oh, that was well played, Humberto. But I
got another chat with him on the 14th. We're gonna do another live chat on the 14th. So
I saw that. Is that with the wax foundation with the wires?
Yeah, we'll touch on the wax foundation, but then I'm gonna do a follow-up to the UBO to give some updates there too. So
you won't want to miss that.
Yeah, come check it out.
Awesome. Kaira, thank you so much.
Absolutely. It's been a pleasure. Thanks for all the work you do. You know, I think like I said, hygiene, I just think it's the future. I think it's
It
the way
is.
to go, this selecting for resistance. And so I really appreciate all you've done for the field, and we gotta get everybody selecting for the best bees out there, and improve everybody's health. The beekeepers, the bees, the native pollinators. I mean, there's so many good reasons to do it.
I'm glad you added that native pollinator thing too, because it makes so much sense. If the bees are healthier, everything's getting pollinated better, so we're eating better. The beekeepers hopefully won't starve to death, because it's hard to make money as a beekeeper. And then adding the native health, or actually how you're affecting everything around you, the ecosystem. I'm glad you mentioned that. That's huge. So I'll
Yeah,
keep
not
that
to
in mind.
mention the hive products and contamination, and there's just so many
Absolutely.
things. Yeah,
But you're right, it is the future. So I'm looking forward to seeing what else you come out with, and I'm ready to see UBO hit the market. I'll be telling a lot of people about it.
me too. Thank you. Can't wait.
Thank you.
We're
Have an
working hard,
awesome
I
day.
promise.
All right, I know you are. Thanks again, Kara.
Sure, I appreciate it, Cory.
We'll see you later.
All right, bye-bye.